Welcome to This Explainer. Today, we're going to untangle the absolutely awe-inspiring process of how a single, solitary cell becomes a fully functioning brain. It's a journey that takes us from the very earliest moments of embryonic development all the way to the ongoing wiring of our adult minds. And we're going to look at the exact techniques scientists use to literally shed light on this invisible world.
Right off the bat, to really understand the brain, we have to ask a fundamental question. How does a tiny cluster of embryonic cells know exactly how to construct the incredibly complex networks that will eventually govern human memory, emotion, and behavior? Without a master builder calling the shots, how does this biological architecture assemble itself so perfectly? Well, let's follow this journey through 4 key stages. We'll start with the birth of the brain, look at how neurons find their way, dive into the genetic tools used to light up the brain, and finally, look at adult brains and new neurons.
Okay, let's jump right into part 1, the birth of the brain and starting the blueprint.
It all kicks off with a process called neural induction. At this early stage, you basically just have a bunch of generic embryonic cells, but then they receive these highly specific molecular signals, instructing them to become the neural plate. This is absolutely crucial. It's the foundation, the very first nervous system tissue from which the entire rest of the brain will eventually emerge. And this induction is quickly followed by neurogenesis. This is the actual generation of new neurons from neural stem cells and progenitor cells. And guys, the sheer scale of this is mind-blowing. In human embryos, this is a period of explosive growth. We're talking about hundreds of thousands of new neurons being produced every single minute at its peak.
Moving on to part 2, finding the right path, or neuronal migration.
Because, simply being born isn't enough. These newly minted neurons must now find their designated places in a vast, developing structure. Through a process called neuronal migration, young neurons have to travel what is, on a cellular scale, vast distances. Early on, they used this fascinating method called somal translocation. Essentially, the neuron extends a long process out to the edge of the developing brain, attaches itself to the surface, and then literally pulls its own nucleus up through that extension and into the emerging cortex. It's kind of like throwing a grappling hook and pulling yourself up.
So the crucial point here is, how do they know where to go? Well, as they migrate, structures at the tip of their axons called growth cones act like microscopic antennas. They sample the local environment for guidance. They follow survival cues like BDNF, that's brain-derived neurotrophic factor, and they react to specific molecules like reelin, which acts as a precise stop signal, telling the neuron exactly where to park and integrate into the cortical layer.
Which brings us to part 3, and honestly, a fascinating plot twist. How do scientists actually see these microscopic, invisible processes happening in a messy biological soup? Let's talk about lighting up the brain.
Researchers use a highly deliberate, structured 3-step process to map the brain. First up, they genetically modify a specific cell type or region. Second, they measure the effect on behavior. So, if the behavior changes, they know that region is involved. And finally, step 3, they use specialized genetic tools to visually identify and map those exact modified cells.
Take a look at this. It's a beautiful, almost pastel-like map of a fruit fly's neural pathways. By using reporter genes like green fluorescent protein, or GFP, scientists can make specific, targeted neurons glow against a dark background. It completely removes the guesswork, revealing the precise location of the neurons that are driving specific behaviors. And this brilliantly illustrates the power of these advanced tools. Now, this isn't just your standard GFP. This is tau GFP. Tau is a protein that binds to microtubules, which are basically the internal skeleton of the cell. So by attaching a fluorescent marker specifically to tau, researchers don't just see a glowing dot for the cell body, they illuminate the entire microtubule skeleton. It reveals the cell's complete structure, including its long, complex axons.
But you might be wondering, how do we only light up the specific cells we want? Well, this targeted visualization is incredibly precise thanks to the GAL4/UAS system. Think of it as a genetic lock and key. It allows scientists to drive these glowing reporter genes only in very specific, chosen neurons. If the key, which is GAL4, isn't present in a particular cell, the lock, which is UAS, won't open, and the fluorescent protein simply won't be expressed.
Another incredible technique is immunohistochemistry. It's a bit of a mouthful, but it is perfect for looking at mixed, heterogeneous tissues. Here, scientists use a primary antibody to target a protein unique to a certain cell state, like nestin, which is found in embryonic neural stem cells. Then a secondary antibody carrying a fluorescent tag binds to the first one. The result? This vivid, color-coded distinction of cell types. You can make neural stem cells glow red and nearby glial cells glow green, allowing for exact mapping and quantification.
By combining all these visual tools with electron microscopy and advanced machine learning, scientists are achieving the ultimate goal, reconstructing the central brain connectome. What we're seeing here is a map of the exact structural connections, or synapses, between thousands of individual neurons in circuits like the mushroom body. They are completely mapping out the structural highways of memory and learning.
Let's move to our final section, Part 4, Adult Brains and New Neurons. Because this illuminated blueprint isn't just for developing embryos, it's a dynamic process that continues throughout our entire lives.
For a really long time, the strict scientific dogma was that you were born with all the neurons you'd ever have. But after a highly heated debate in the 1990s, researchers definitively proved that the fully developed adult brain does indeed produce new neurons. And they did this using a brilliant tool called BrdU. For anyone not in a lab coat, think of BrdU like a microscopic chemical tracker. When cells divide to create new neurons, they have to copy their DNA. Scientists figured out how to trick the brain into using this BrdU chemical as a building block for that new DNA. Later, they can apply a wash that literally makes the BrdU light up. This gave researchers definitive visual proof of exactly which neurons were newly born. This incredible process, known as adult neurogenesis, happens predominantly in a region of the hippocampus called the dentate gyrus.
And thanks to that BrdU tracking, we know it's not some insignificant number either. In fact, an astonishing estimated 1,400 newborn neurons are generated in the human hippocampus every single day. Your brain is quite literally continuing to build itself right now as we speak.
So what do these 1,400 daily new neurons actually do? Well, they mature and functionally integrate into your established neural networks. And importantly, they show a remarkably high sensitivity to long-term potentiation, or LTP. LTP is essentially the cellular mechanism for learning, meaning these fresh neurons are hyperreceptive to forming new connections. They play a critical role in spatial memory and active learning. Conversely, when this process of adult neurogenesis is defective or impaired, the consequences are severe. Deficient neurogenesis is strongly implicated in several major disorders characterized by defective learning, memory, and mood regulation, including Alzheimer's disease, schizophrenia, and major depression. This highlights exactly why studying this biological blueprint matters so much to us all. Which leaves us with this final thought. As we continue to refine our ability to literally light up, map, and understand the origins of our minds, from the embryonic neural plate to the adult hippocampus, what revolutionary treatments might this illuminated blueprint unlock for the future of human medicine? Thank you for joining me for this explainer, and keep questioning the incredible machinery inside your own head.